So thank you for coming to my talk today. My name is Emma Lehmberg, and I am a postdoc at the University of Toronto. But for this talk, I'm going to be discussing my PhD work, which focused on examining how sexual conflict can act to reproductively isolate lineages over macroevolutionary time. So if you've been sitting in this session before this one, or you happen to study sexual conflict, you will know sexual conflict is when males and females have differing fitness optima when it comes to reproduction. And these differing fitness optima can drive antagonistic coevolution, and if strong enough, eventually reproductively isolate populations from one another. So here you can see two ducks. The male might want to father as many offspring as possible to guarantee parentage, where a female wants to control access to her gametes to ensure that she has the best possible offspring, and they have the best possible chance of surviving. Sexual conflict can play out at any stage during mating or interaction between the sexes. So there is kind of a continuum of interactions, but I'm only going to mention 3 of these arenas today, and I'm only really going to focus on one, which is post-zygotic. So pre-zygotic is anything that leads up to mating. It can be the evolution of morphologies that give an advantage to one sex over another, and then the opposite sex kind of responds by evolving an avoidance mechanism. Post-mating pre-zygotic is anything that happens after the act of mating, but before fertilization, so it can be sperm competition. And then post-zygotic is anything that happens after fertilization or involved in the care of offspring. I'm particularly interested in post-zygotic sexual conflict, specifically viviparity. In viviparous animals, there is evidence that interlocus sexual conflict, or conflict that involves many loci across the genome, can accelerate lineage isolation and diversification via this physical connection between mother and offspring. And it can provide an arena for sexual conflict to play out after fertilization. So it continues on. And here you can see this figure, which demonstrates, so on the y-axis, you're seeing the number of successful hybrid pairs that exist in these different taxa. And the thing that you're immediately going to notice that in mammals, which you may know are viviparous, the ability to hybridize successfully, so this would be like producing offspring that can then mate with other individuals or have the ability to do that, it disappears over time much more quickly in mammals than it does in birds or frogs, which are egg-laying, as you also may know. In reflection of this particular pattern, Zay and Zay proposed the viviparity-driven conflict hypothesis in 2000, and then they followed it up with another paper that sort of elucidated on it in 2008. But basically this says that intra- and intergenomic sexual conflict result in sort of this perpetual antagonistic coevolution, which can accelerate interpopulation post-zygotic isolation. So it can provide this initial barrier to producing viable offspring. And so we know a decent amount, not enough, but we know a decent amount about how this might operate in microevolutionary scales. But it's a little more difficult, and there's a little less work done on it in sort of a macroevolutionary time scale. And so one way we can actually study the sort of reproductive isolation as a reflection of sexual conflict is to look at patterns of hybridization through time using a phylogeny in relation to traits that have evolved as a result of sexual conflict. So there are a couple of requirements that need to be met in order to be able to do this. One of them is that hybrid offspring need to be fertile, they need to be able to reach adulthood, and they need to be able to reproduce with parent species, so that you can then have the passage of genomic material from one species into another. You also need to be able to retain, to a certain amount, that sort of genomic material that's been passed to another species. And so the variability in ability to retain, sorry, that's a complicated phrase, inability to retain that hybrid genome that you've acquired as a species can reflect sort of selective events or selection pressures that are happening within the genome through time. And so, just a note, I'm going to use introgression and admixture interchangeably in this talk. They do have slightly different definitions, but for my purposes, we're just going to assume they're the same. So, and actually, let me just talk about this slide for a second. So this is a phylogeny that was published in 2013 by Sui et al. And it shows hybridization through time across the genus of Xiphophorus, which you may know commonly as the swordtails. And why do I bring this up? Because I am talking about the Poeciliids, which is the family that this genus comes from. It's a taxon of about 275 putative species, possibly more. There are a lot that have yet to be fully described. And it's a radiation that's about 50 to 60 million years old. The whole family possesses a state of live bearing, but the type of live bearing differs, and this is where things get interesting. They also, in at least the species and genera that have been studied, have this extensive history of hybridization. So they provide this really great model to look at how traits of sexual conflict sort of influence, can guide the reproductive isolation of this group. So just to give you a breakdown of the types of live bearing in this group, there is a spectrum, so it's not either or. It can be along this line. You can be sort of one or sort of the other, but I'll talk about the two extremes to keep things simple. So on one side, you have placentation, which I will refer to as matotrophy for the rest of this talk, and that's where there's a physical connection between mother and offspring as they develop. There's very little yolk. On the other end of the spectrum, you have leucithotrophy, which is far more common. And in this case, it is a fully yolked egg that is carried to term within the mother's body, but there's very little interaction between mother and offspring. Interestingly, there are male traits that correspond with these female reproductive modes. So here you can see a matotrophic species. Males tend to mate coercively, so there's no precopulatory mate choice that we know of in a lot of these species. And they inseminate her with a modified anal fin called a gonopodium, which is present throughout the entire family. They're often smaller than females, and they're monochromatic. Conversely, we have leucithotrophic males, which court. They're big, they're beautiful. They have these flashy ornaments, and they tend to be dichromatic and larger than females, so there's this suite of traits that accompany these different live-bearing modes. So given all these things, we wanted to ask a few questions. So the first is, do matotrophic clades exhibit less introgression than leucithotrophic clades, reflecting different degrees of sexual conflict that are occurring? And then secondly, are pre- or post-zygotic traits of sexual conflict more predictive of admixture? So to do this, we sequenced whole genomes of 204 Poeciliids, put them through this pipeline, which I'm not going to talk about very much because I think that people are pretty familiar with it. If you would like to talk more, please find me after. We use the variant culling files to reconstruct chromosome-level coalescent phylogenies with SVD quartets, and then we use these as a backbone to sort of calculate discordance. And then we also isolated phylogenetically informative SNPs, and then we I broke them down into 50 kb SNP windows, and then we reconstructed maximum likelihoods with IQ tree. We also calculated gene and alignment site discordance. This is just something to give us a measure of how different these window trees are from each other across the genome. And then we also calculated two metrics of admixture. So one is the F4 ratio. It just tells you how much proportion of the genome each species has that does not belong to itself. And then we also calculated an F bench, which we modified slightly, and I can talk a bit about after the talk if you would like to know how we did this, to calculate sort of an extant species admixture. Okay, so what did we find? So here's the phylogeny. If you happen to be a Poeciliid person, and I'm only going to talk about two genera in this talk. If you happen to be a Poeciliid person and you want to talk more about this, please find me. I am psyched about this phylogeny, but I don't have time to really focus on it. So what you're seeing here is each tip is labeled by the live-bearing state, but then you also have concordance factors, and the thing that becomes immediately evident from this phylogeny is that there's an incredible degree of variance between genera that have different live-bearing modes across the phylogeny. A different way to look at this is simply to plot it as a violin plot and look at how many matotrophic species are in each major genus. So I'm only focusing on major genera here, plus one that's wholly matotrophic. And what you see is as you have more matotrophy in the genus, you tend to have more concordance across their little clade or large clade. The exception here is Philoceras, and this is because there's a large polytomy in the clade, so it was really hard to figure out what exactly is going on. It's a recently diverged genus, so there's probably a lot of other things going on here, and we haven't really looked into it yet. However, this is telling you about discordance and not necessarily hybridization. So we also looked at these F4 ratios. So we're just comparing pairs of species and their admixture. So this is a largely matotrophic genus called Pachyliopsis. And what you can see from the phylogeny is there's very little discordance. And this heat map, which I'll orient you to, so the darker the color, the higher the support for evidence of admixture. The warmer the color, the more admixture you're seeing. And the admixture is on a scale from 0 to 0.3. There's very little in this group in comparison to this one. So this is a wholly lithotrophic clade. There is a lot of discordance in this clade. We know from previous studies and a lot of the work that's been done in Molly Schumer's lab that there's a lot of hybridization happening here. And so it's interesting to see these patterns replicated and approaching the question in different ways. So yes, introgression is more prevalent in lithotrophic fishes with no placental connection between mother and fetus. But we also wanted to see, okay, so we have these traits that are associated with modes of live bearing. Are they predictive? Which type is more predictive? So in order to do this, we took a pretty straightforward approach. We calculated phylogenetic least squares for every single tree and every single trait in relation to admixture across the genome, which generated a sort of a distribution of relationships. So this is not just one tree we're using, it's the whole genome's worth of trees, which is great, but it also makes things slightly more complicated. Our prezygotic traits were gonopodium length, sexual size dimorphism, which is a ratio between male and female body size, sexual diphenism index, which I'll talk a bit about more in a second. And then our postzygotic trait was literally just looking at the matotrophic index, which is a calculation of how much connection there is between mother and offspring. So the sexual diphenism index is just looking at phenotypic traits of males. And so it's composed of ornamentation, dichromatism, and courtship. And so if you have all three of these things, you have a score of 3. If you have none of these things, you have a score of 0. And if you have some but not others, you're in between. So it's sort of a simplified way to look at this. So what did we find? 19.7% of the window trees show a significant relationship between SDI, the sexual diphenism index, and introgression, which is not a lot, but what is interesting is when you calculate the variance, which you can see across each category at the top, it's almost 4 times the variance of admixture, sorry. It's almost 4 times what it is in the first category versus the last. So remembering that if you have a score of 3, you court, you're dichromatic, and you probably have some cool ornaments. For the sexual size dimorphism, this is more significant, and so 48% of the trees have a significant relationship between this particular prezygotic trait and admixture. And just to give you an idea, I don't have it on there, never mind. There's no generic pattern. And then for relative gonopodium length, this is probably the strongest relationship. There's 57% roughly of these trees shows a relationship between the gonopodial length, which is longer if you are more matotrophic and so mate more coercively with admixture. And so just to give you an idea of where our example fish are, Xiphophorus tends to have short gonopodia, and Pachyliopsis tends to have very long gonopodia. And then finally, our postzygotic trait, the only one that we looked at for this, is perhaps the least predictive of admixture. And I actually think this has more to do with where loci are found in the genome. So as of yet, we haven't identified where the conflict is coming from. And so I think if we subset these trees, we'll find some interesting things. We just haven't done that yet. On the whole, blah, 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 conclusion, next person. So come find me.
So it's 11:30. I think the chime kind of comes a little late in this room, but it's 11:30, so I'm going to get started. I would first like to start by acknowledging my co-author on this work, Isabella Martinez. Izzy was an undergraduate in my lab for 3 years. She just graduated in May, and the work that I'm talking about today was her undergraduate senior thesis research that she was awarded a Sigma Xi grant in aid of research to complete. So there's a tremendous amount of variation in mating duration in insects. I'm going to talk about a couple of extreme examples. On the one hand, we have the yellow fever mosquito, Aedes aegypti, that can mate for as short as 13 seconds. And then on the other end of the spectrum, we have the southern green stink bug that can stay in copula for as long as 10 days. So this is a remarkable amount of variation between species, but we also see variation in mating duration within species as well. In the mosquitoes, matings can range from 6 seconds to over a minute, and matings in the green stink bugs can last for as little as 5 minutes, raising questions about why there's so much variation in this trait and why matings last for so long. Especially because in many insect species, sperm are not transferred continuously throughout matings. So longer matings do not always translate to higher numbers of sperm transferred. So in species where sperm transfer and mating duration are not correlated, why do we see these prolonged matings? Particularly because they can be costly. If you're a green stink bug in the field mating for 10 days, this is going to increase your predation risk. But even if you're, say, a laboratory organism, longer matings are energetically expensive and can use up time and energy that you could otherwise be directing towards other behaviors. In species where mating duration is under female control, this could be a form of cryptic female choice, or it could provide protection to females from additional male harassment. In species where mating duration is primarily under male control, we typically see prolonged matings associated with increased success at sperm competition for males. And the mechanisms behind this vary in insects and can include longer matings allowing males to transfer higher quantities of seminal fluid proteins, allowing for the formation of mating plugs, or prolonged matings can act as an extreme form of mate guarding in these species. I work with an insect that also has prolonged matings, the fruit fly Drosophila melanogaster. In the species, mating duration is primarily under male control, and it lasts much longer than is necessary for sperm transfer to occur, 2 to 3 times longer after sperm transfer has ceased. There's a remarkable amount of behavioral plasticity in this trait in melanogaster. Males will alter how long they mate with a female based on their mating history or the mating history of the female, based on their past or present social environment, and based on the size of the female that they're mating with. And most of the investigations into the consequences of variation in mating duration have used this sort of plastic variation in this trait. But even in this model system, controlled experiments that are sampling just the intrinsic variation in mating duration in a population are lacking. And that's one of the things that Izzy wanted to look into. So we use a tool in my lab that can allow us to sample this intrinsic variation. Hemiclonal analysis is a technique that was developed by Adam Chippendale and Bill Rice in the early 2000s. And it takes advantage of some of the genetic tools and constructs available in this model system. I'm not going to get into details, but essentially what this allows us to do is isolate and clone sperm that we use to fertilize variable eggs, producing a hemiclone line in which males are essentially wild type, but genetically identical at half of their genome. And then we can use this process over and over again to sample additional sperm, clone them, and produce multiple copies of hemiclones that we can then test for a trait that we're interested in to sample the variation in a population. So during her junior year, that's what Izzy did with a then senior in my lab, Grace Freed. Together, they measured mating duration for 29 different hemiclone lines, and they found substantial standing additive variation for this behavior. This isn't the first time this result had been shown. We expected to find this. This has been shown in several other studies. But for the purposes of our research today, what Izzy and I focused on was the 3 lines in which males had the longest mating durations and the 3 lines in which males had the shortest mating durations. And we used these 6 lines to ask whether this variation in male mating duration has any consequences for male or female fitness. And we wanted to try and set these experiments up with as much experimental control as possible to try and minimize the environmental influence that we know can affect this trait. So we controlled the mating history of both sexes, we controlled the social environment of all of the experimental flies, and we controlled the body size of the females that the males were mating with. So I'm first going to tell you how we controlled female body size, and then I'm going to show you why I think this is important. We used well-established methods in Drosophila melanogaster to manipulate female body size by altering larval density. So we created large females by placing 50 eggs into vials containing 10 milliliters of food, and we created small females by placing 100 eggs into vials containing 1 milliliter of food. And past work from my lab has found that the large and small females that we create using these methods have body sizes that fall within the normal range of body sizes found in our laboratory population, and that males court and mate with these females as they do females that haven't had their body size altered. So this represents a relatively high throughput way to control female body size for these male mating experiments. So let me show you why I think this is something that's important to do. What we're looking at here are the mating durations for these 3 long and short mating lines when they're paired with large and small females. So we've got mating duration on the Y and female size on the X. And the first thing you can see, which is great, is that these long mating hemiclone males from these long mating hemiclone lines consistently mated longer, regardless of the female size, than males from the short mating lines. So this is a behavior that is consistent across the range of female sizes. But the other thing that you'll notice is that both long and short mating males mated for significantly longer with larger females than they do with smaller females. Again, this isn't a new result. We knew this. We expected this going in. But I think that these results really highlight the importance of considering female body size when we're trying to understand the fitness consequences of variation in mating duration. So the first thing that we wanted to do was look at a male fitness-related trait, and we measured male defensive reproductive success. And the way that we did this was a 2-part experiment. We first mated these long and short mating hemiclone males to both large and small females. And for this experiment, the females were created in an outbred population that is homozygous for a recessive brown eye mutation. Remember that the hemiclone males are wild type and red-eyed. So we paired these males and females, and then we watched them all to ensure that they mated, recorded their mating duration, and then when all of the matings had ended, we removed the mated females and transferred them into a remating vial that contained a competitor male from this brown-eyed population. We left the females there for 6 hours to give them the opportunity to remate with this male, at which time we removed the males, and we gave the females another 18 hours to lay eggs. And then 12 days later, we counted their offspring. And this is where using the females and the males from this brown-eyed population is so advantageous, because any brown-eyed offspring that the females produced had to be fathered by that competitor male, indicating that the female remated in this vial, and any red-eyed offspring had to be fathered by the long or the short mating hemiclone male during that initial mating. So we then measured male defensive reproductive success as the proportion of offspring he sired across all of these females, and we compared this metric between our long and our short mating males. I'm going to present our results in the same way that I did for the mating duration. We have defensive reproductive success as a proportion on the y-axis and female body size on the x-axis. And when we got some really interesting results. So I'm going to start by focusing on sort of the right side of the graph with these smaller females. And what you can see here is that the males that had longer mating durations consistently outperformed the males that had shorter mating durations with these females. They had much higher defensive reproductive success than the short mating males. If we look at the large females on the left, however, we see no difference, no effective mating duration on male defensive reproductive success. So it appears that the benefits that males can get from these prolonged matings can depend on the body size of the female that they're mating with. Now, that said, I do expect that these benefits are going to be relatively common in our population. I told you that both these large and small females have naturally occurring body sizes in our population, but the large females really represent like the upper echelon of female sizes we get in our population. Only about 5 to 10% of females in our population have body sizes as large as the large females we're using, whereas about 40% of the females in our population have body sizes consistent with the smaller females. So we do expect that males will likely receive benefits from prolonged matings across multiple females in this population, providing a fitness benefit to males associated with these prolonged matings. And this isn't really surprising because we know that mating duration is primarily under male control, so we would expect there to be some sort of benefit to males if they're exceeding the matings beyond sperm transfer. But we weren't sure what would happen with the females. So we also set up a female fecundity assay. This is set up very similarly to our male assay, except for this experiment, the females came from our just outbred wild-type population. Again, we paired them with long and short mating males in all possible combinations and watched them to make sure they mated. When the matings had ended, we removed the mated females and we placed them into an oviposition vial for 24 hours. And then we measured female fecundity as the total number of offspring the females produced in that vial after their mating, and we compared this metric between females that had mated to long and short mating males. The first thing that you're going to see when you look at my data is that the small females had substantially lower fecundity than the large females. This wasn't surprising. In Drosophila, like in other insects, female body size and fecundity are positively correlated, so we were expecting this result. But what I want you to realize is that that blue line for the long mating males is consistently below that orange line for the short mating males. And in fact, both the large and small females had significantly lower fecundity after mating with males from these long mating hemiclone lines compared to males from these short mating hemiclone lines. So these longer matings are actually costly to females because they're reducing their fecundity. So taken together, the fact that longer matings increase male fitness and decrease female fitness indicates that there's sexual conflict over this trait in Drosophila melanogaster, at least in this population. But we don't yet know the mechanism that's driving this conflict. A beautiful hypothesis is that males are using these prolonged matings to transfer higher quantities of seminal fluid proteins like we see in other insects. And this would be consistent with some of our results because these proteins can delay female remating and increase male success at sperm competition. Although they can rev up female fecundity, they are also toxic to females, so that might explain what we're seeing with the females. But studies in this species looking for a relationship between the quantity of seminal fluid protein transfer and mating duration are mixed. So I don't think it's clear that this is necessarily what's operating. And there are a couple of other possibilities that I'm sort of vaguely calling mechanical mechanisms. We know that females will eject sperm after mating in this species, so maybe these prolonged matings can interfere with that process in a way that benefits the males. And then from the females, I think we should potentially look at the way in which copulation actually happens in this species, where, as in other Diptera, the male mounts the female, and then sort of there's some genital wounding involved, and then he pretty much rides around on the female for the duration of mating. So if these matings are energetically expensive or damaging to the female, it's possible that longer matings exacerbate these costs, and that could explain the depreciation in fecundity that we're seeing. So regardless of the mechanism, we do think that mating duration is involved in sexually antagonistic coevolution in this species, in which males will experience selection to lengthen matings even at a cost to female fecundity, and females will experience selection to shorten matings, even if this is at a cost to the males they mate with. So we think that sexual conflict could be contributing to some of this variation in mating duration that we see within this species and potentially other species of insects. So with that, I would like to just thank all of the undergraduates who contributed to this work, and I'd love to take any questions.
So my name is Kyndall Skelton. I'm a first-year PhD student at Auburn University, and today I'm going to be talking to you about quantifying the routes by which natural selection is shaping sexual size dimorphism in C. maculatus. So sexual size dimorphism is very prevalent across a wide range of taxa, impacting almost 90% of organisms that have different sexes. So this is not just in males, as you can see in this wonderful graphic that was drawn by Dr. Robert Cox. Sometimes it's in females as well, just like in this native island plant to New Zealand, where the female is larger than the male because she needs to carry seeds. And also in this praying mantis, who lays up to 200 eggs at a time, and she also has aggression towards her mate during her mating period. And in anoles, the males are bigger than the females, because he's aggressive towards other males of his species in order to get more mates. But that is not always the case, and movement away from size equilibrium can occur to several different factors, including competition for resources, enhanced reproductive roles, or even improving survival rates. So what happens when these traits are evolving independently due to sex-specific selection pressures? So to talk about this, I'm going to look at C. maculatus, like the title of this talk said. And so females are larger than males in C. maculatus because females need to produce and store more eggs, which leads to greater fecundity, and males are smaller, so they reach reproductive age quicker, and so this has more mating opportunities for them. So C. maculatus, the males and the females do share a majority of the same genome, so this can lead to difference in sex-specific gene expression and regulation, reflecting interlocus sexual conflict where a trait that's beneficial to one sex may be detrimental to another. So looking at this figure on the right, the top panel shows no conflict at all. The bottom left panel shows partially resolved conflict where the sexes are constraining each other, and so some of their needs are being met, but not all of them, and then fully resolved conflict where each sex has their own specific optima. And so sexual size dimorphism is typically measured with a univariate approach, where you're looking at one trait and its effect on the trait of interest. And But in this talk, I'm going to talk about moving more towards a multivariate approach where multiple traits can look at the direct and indirect effects on fitness and, yeah. So some relevant information about C. maculatus. So C. maculatus is a widespread pest, also known as a cow pea weevil. It's found on every continent but Antarctica, and it typically infests bean stores, which it makes it really easy for us to emulate their natural environment within the laboratory setting. And the previous findings that are important to this talk is that there is that positive size and fecundity relationship. So bigger females are having greater fecundity, and shorter male development time is reducing that interlocus sexual conflict. Now, if you know anything about studying the strength and nature of natural selection, it's often challenging because it requires extensive fitness data from many individuals within a population. Well, lucky for us with our seed beetles in the lab, we have reared like 25,000 over replicate 1. It's been a lot. So to tackle this, we took advantage of this ability to rear so many in such a quick period of time, and their evolutionary history and a great bit of knowledge about the sexual conflict in the system to develop an experimental plan that allowed us to measure not only size but various different traits. So our experimental design, we mated individuals monogamously in the lab, that we put them on bean dishes for 48 hours. After this 48 hours, they were removed and the hatched eggs were sorted to create the new generation. So each individual that emerged from the new generation was weighed as an adult upon emergence, and then they were either put back into these monogamous matings or they were put into our reproductive success assays, which creates our hatched egg counts. So what exactly is a reproductive success assay? So we had 1 focal individual, 3 same-sex non-focal competitors, and 4 opposite-sex mates. Our 3 same-sex non-focal competitors were sent to the cobalt-60. If you're not sure what this is, it's a source of gamma radiation, which is used to sterilize non-focal individuals. So then all 8 of those individuals are actually put into a bean dish together, and again, 48 hours. And then they are all removed, and after the maximum hatching time, the bean dishes are counted with 2 independent counts to give us our reproductive success measure, which is number of hatched eggs. So utilizing this component of how many beetles were put through this process, it was roughly 900 males and 900 females across 8 generations total, with, here's the distribution of the number of hatched eggs. Males had around 160 for their max, and females had around 90. And as you can see, the males did have a higher variance than the females did. And this is this is due to the fact that absolute reproductive success is the opportunity for selection in each sex. So the males had more egg-laying individuals in their dish than the females did. And we use this number to calculate our relative reproductive success. And this was done per generation, per sex, rather than across the entire population. And this is just the individual number of hatched eggs divided by the generation per sex, number of hatched eggs mean. And so the expected mean for this was 1, which you can see in the distribution. It is centered around 1, but there was a lot of variance. It does extend all the way out to 4. So how did we use this metric of relative reproductive success to tell us how selection is differing between males and females when sexual size dimorphism is present? We used the Landon-Ardell 1983 selection analysis approach to build univariate models, multivariate models, and correlational selection gradient models to kind of assess how the selection gradients on one trait might be correlated with one another, and also trying to reveal the complex interplay between traits under selection. So what did we find? So our population is sexually size dimorphic. As you can see, the males are 6 milligrams and the females are around 8 milligrams, and they have their own peaks that are apart, as you can see. This graph was done with raw values, and everything else is done with scaled and standardized values. So the males are experiencing positive directional selection and stabilizing selection, so larger males are being favored in terms of reproductive success, possibly due to increased competitiveness or enhanced mating opportunities. However, the stabilizing selection is saying that these extremely large sizes are not being selected for. And the same was found in females. Now, the larger females being selected for was something we were expecting because of that increase in fecundity. However, at a certain point, the larger sizes seemingly are selected against as well. This is probably due to some energy or resource required to maintain a very large body size, which could offset that benefit of increased fecundity. We did find something really interesting, though, which is that there was significant quadratic selection differing among the generations whenever we looked at it all together. Now, since the values are centered around 0 and it is fairly weak selection, if we were to continue this for, say, 100 generations, we may get the same result, indicating noise, or this could be evidence that natural selection is not consistent throughout time, which is an outstanding question currently in our field. We also looked at juvenile development time. So this is also done with raw values, and the males and females are very similar. This was done hourly from 8 a.m. to 3 p.m. on days of emergence. That's what the gaps in the distribution are. So males were experiencing negative directional selection, which, as I talked about earlier with the males with shorter development times, is resolving the interlocus sexual conflict. That'll come back later. But so the males that are emerging earlier are having higher reproductive success. This is probably due to more mating opportunities, whereas the females had no significant selection at all on juvenile development time within our multivariate analysis. However, the females did have significant selection occurring within our univariate analysis, so this could be indicative of a direct impact of juvenile development time on reproductive success. However, the impact is probably through an indirect relationship with other variables such as mass and emergence rather than having a true direct effect, which is something interesting that we did find. Now, age. So the males are experiencing directional selection on age negative and stabilizing selection, meaning that there probably is a optimum mating age for males in our population, and the females were experiencing negative directional selection, so just the younger ages were having higher reproductive success, but as you can see from these graphs, the ages didn't really change that much on the scale. So there was differing directional selection among the generations. Like I said with the other one, if we were to continue this, it could just be noise, or it could also be our population is experiencing evolving reproductive strategies or environmental changes. We're unsure right now. So what does this mean for where we are? So to kind of recap, both of the sexes want to be heavy but not too heavy. The males are emerging earlier. Mating age is, both want to be younger, but males, there seemingly is an optimum age for mating. But in both sexes, mass at emergence is subject to positive directional selection, as you can see in this graph, kind of. It's a little confusing. There's a lot going on, but yeah, they are color separated. So the strength of selection is similar across both sexes in mass and emergence. This suggests that the selective pressures driving size increase are probably consistent, and as a result, sexual dimorphism in size is likely to be maintained. There's not a ton of curvature in these splines, so the directional selection is, or makes it seem like there isn't sexually antagonistic selection occurring, but at the same strength, so sexual size dimorphism should not decrease. That being said, we can bring back this lovely graph down here in the bottom. It's smaller now, but we can bring this back and look at it, and where are we? So initially, our lab hypothesized that sexually antagonistic selection on mass might stem from indirect selection on size, driven by direct selection on key life history traits like juvenile development time, which differs between the sexes. However, our data did reveal the opposite. Juvenile development time had a weak impact on size, and yeah. So with that, I kind of flew through that, I'm really sorry. But with that, I'd like to thank the members of my lab that worked on this project. We had a lot of amazing undergrads in the Auburn University Physics Department who helped us with the Cobalt 60. Would not have been able to do that without them. And our funding, and Dr. Charles Fox at the University of Kentucky, who without him, we would not have our beetles. And with that, I can take any questions.